Good afternoon, class. Welcome back. The remainder of today's session and continuing on to next week's session, we're going to deal with the subject of microbiome. In the preceding lecture by Dr. Purnima Bhanod, you focus more or less on specific aspects of bacteria, namely their role in human pathogenesis and the tools available to us for controlling specific bacteria. But our interaction with the bacterial world is far more extensive than the roles of individual bacteria in causing human disease. In fact, most of those interactions are beneficial to us as well as to all other organisms on this globe. So we're going to start with, in part one, on the definition of what a microbiome is, what its composition is, how we determine its composition and its function, and as well as the roles of the microbiome in normal human beings. In the second half, we'll deal specifically with the roles of microbiome in human health situations, including a number of diseases. All right, let's begin with the first slide. Up until the onset of the pandemic early last year, the one topic that dominated medical news, for example, toward the end of 2019, was the microbiome. Some of what was happening is shown in the top left of this slide. This simply is a graph showing the number of publications, microbiome citations that showed up on PubMed, which is the main repository of biomedical information in the world. You can see that it took off around 2010 and it has been increasing exponentially up until 2016. This line goes way up beyond the screen if you were to extend it to 2020, for example. What happened here? Why did it go up suddenly? Did we recognize something that we didn't know before? The answer is yes and no. But concomitant with this explosive growth in literature, with, of course, interest generated in the popular press, New York Times articles, for example, but also alleged cause-effect correlations between various human conditions: allergy, Crohn's disease, autism, immunity, obesity, cardiovascular disease, diabetes. And there were articles on gut-brain axis and so on and so forth. And invariably, of course, there were also quickie books that try to implicate microbiome in various human conditions. Some were snake oil proponents, and others were more serious. Nevertheless, microbiome came to dominate medical news up until the onset of the pandemic. Now, the pandemic is going to be controlled eventually, and the importance of human microbiome is going to return to the center stage. Let's go on to asking first, what is a microbiome? This word was defined by Joshua Lederberg, who was sort of the grandfather of bacteriology. In 2001, he defined it as an ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. A couple of nomenclature issues I would like to draw your attention to is the word microbiome refers to genomes of microbes from a community. Okay, so this is referring to the genes, right? Microbiota refers to the organisms. It's a subtle difference, but something that you should bear in mind. All right. A microbiome has many types of microbes. A microbiome does include bacteria, archaea, fungi, viruses. However, most research has focused on bacteria. That does not mean that the other organism types, such as archaea, fungi, and viruses, are not important. They are assuredly important. It is just that we don't know as much about them as we know about true bacteria or eubacteria. Okay? Something else all to bear in mind. Okay. Now, there are many different human microbiomes. Sometimes they're also called quote unquote normal flora. The most important microbiome in the humans is the one in gastrointestinal tract, but there are other important ones. For example, vaginal, respiratory tract, and skin as well. If you look at the diagram, you have to memorize nothing in the diagram. Just take a quick look at the color scheme used to indicate different phyla that are represented. Clearly, what is in the nose, in the nasal cavity, has a pattern that's very different from a nearby space, mouth lining, for example. Completely different bacteria are occupying these two nearby niches. And both are very different from what is in the gut or what is in the vagina. So there are many microbiomes on a human organism. Okay? All right. Now, now we want to ask, is there any place safe from the microbiome or which is shielded off from microbiome in the human organism? Yes, there is. There are spaces that are sterile normally where organisms are not seen except under disease conditions. And those examples of those tissues are blood, cerebrospinal fluid, many different body fluids, deep tissues, etc. There are other sites in a human organism that are nearly sterile. In other words, they may transiently see bacteria, but not constantly. For example, bronchi, alveoli in the respiratory tract, esophagus, urinary bladder, uterus, all of these may see organisms from time to time, but they're also considered to be nearly sterile. All right. Let's now put microbiome in perspective in this and a couple of other slides that are coming up. The first thing we need to understand about microbiome is that microbiome is the foundation of the biosphere. All multicellular organisms embed their own microbiomes, and in turn, they're embedded within microbiomes, environmental microbiomes. So, multicellular organisms can be viewed as supraorganisms with hologenomes that consist of genomes, epigenomes, and microbiomes. Okay? To give you an idea for the dominance of the microbiome on the biosphere, look at this cartoon version of evolution of life on Earth, right? Bacteria were formed nearly 4 billion years ago, the earliest occupants of the globe. In relative terms, mammals came into existence only about 200 million years ago. Okay? So we came into the world very, very late to the party that was already fully dominated by microbes, specifically bacteria, right? And all life forms we see today that are evident, most life forms were relatively of recent origin relative to bacteria. So it's not surprising that we have such an intimate connection with the bacterial world. Okay, in this slide, I want to draw your attention to the concept of hologenome and holobiont. Okay? These two diagrams on the left side and the right side are somewhat redundant, but I'm using them to drive home the point of what a hologenome is and what a holobiont is.
Dr. Humayun?
Yes.
I'm sorry to interrupt you, but there is a question in the chat.
Okay.
Is the placenta or the womb considered sterile?
Yes, they are considered to be sterile, but there are conditions where there are transient occupation by organisms. Yeah.
Thank you.
Okay. So focusing on the left-hand genome, we'll look at the definition of what a hologenome is. A hologenome consists of the host genome, here we see examples of either a human or a plant, okay, and its own microbiome. Together, they are called hologenome. The hologenome is embedded in an environmental metagenome. In general, environmental metagenome doesn't directly contribute to the hologenome, but the host genome and the microbiome do interact with each other. Okay? That's why we call them a hologenome. Now, the concept of a holobiont is, of course, here the emphasis is shifted to the organism. The host organism and the microbiota carried in the host together are called holobiont. The organisms are called holobiont. Now, according to some evolutionary biologists, the evolutionary unit is the holobiont, is not the individual organisms. We, according to these biologists, human evolution should be viewed not specifically as the human organism's evolution, but more the evolution of the holobiont of which a human is a part. One important point I would like to draw your attention to in this slide and in one or two other slides coming down the pike is the idea of the relative contribution of genes that regulate the holobiont. How much is being contributed by the host? How much is being contributed by the microbiota? Some numbers are given here. Different sources will give you slightly different numbers, but the proportion is the same. The human genome contributes 25,000 genome, 25,000 genes, whereas the microbiome contributes an astonishing 9 billion genes. So clearly the contribution, genetic contribution of microbiome is much, much larger. And from this, you can derive the significance of the contribution made by microbes to adaptation under various circumstances. Okay, now we'll move on to the topic of how do we know what is in a microbiome? In other words, the composition of a microbiome. And secondly, how do we know what they can do functionally? Okay? This, in this slide, we'll also consider explanation for why microbiome publications took off in 2010 and are growing at an exponential rate. What happened? It turns out that historically we did know that there were an immense number of bacteria, especially in the gastrointestinal tract, but most of those bacteria were not cultured. They were in fact not culturable, and they constituted some sort of a dark matter that was beyond reach. We knew there was something there. We didn't know what was there because we couldn't culture them. All the genetics of bacteria up until recent decades was based on isolating individual colonies and characterizing individual bacteria. We had no tools to analyze populations of bacteria which were not culturable, which were present in large numbers. So what changed was the genomics revolution, which was seeded by the Human Genome Project, which in turn generated two vast new resources. One resource was simply databases that allowed us to compare any new sequences to what is already characterized, what is in the databases. The other is high throughput sequencing technologies, as well as other computer tools necessary to very quickly accumulate and compare sequence information. Okay, so basically, when you approach a microbiome, you have two major questions. Who is there? Identifying the main phyla present in microbiome is done by a technology called 16S ribosomal RNA or rRNA sequencing, and organizing the sequencing information into operational taxonomic units, OTU. This is roughly the same as a species, what we used to call species. The second important question is, what can they do? What they can do is basically functional characterization is carried out by omics approaches. The most important of them or random sequencing and data mining, that is metagenomics, and transcriptomics, that is which genes are being expressed, and metabolomics, what metabolic products are being generated by this microbiome. Okay? There are other experimental approaches that have been used for functional characterization of microbiome. Those are the use of germ-free animal models and a technology that's very simple but quite effective, microbiota transplantation. So let's visit some of these topics in subsequent slides. Okay, the classification of organisms were done over the last several centuries through various different schemes. However, the scheme that is dominant today for the last 40 years, and the one that's the most universally accepted, is the one proposed by Carl Woese, who was working in the Midwest in University of Illinois in the 1970s. He determined the 16S ribosomal RNA genes from the organisms that were already known and characterized at that time, and with the hope that he would be able to use this highly conserved gene sequence as some sort of a molecular barcode for classifying organisms. And sure enough, he came out with a classification scheme that was able to fit in all organisms whose 16S ribosomal RNA sequence could be determined into just three categories that we know today: eubacteria, true bacteria, archaebacteria or archaea, and eukarya. Okay? This is the scheme that is dominant today in classifying all living things. And that so-called tree of life is shown at the bottom left here. We begin with the common ancestor, which splits into two branches. One branch gives rise to true bacteria, the eubacteria. The other branch splits in turn into archaebacteria and eukarya. This is the scheme that was brought into existence by Carl Woese, and this is the significance of 16S ribosomal RNA historically. Now we go to asking the question, what exactly is the 16S ribosomal RNA and how is it used for determining the microbiome composition, which is bacterial diversity and relative abundance? Now, 16S ribosomal RNA, which is shown in a folded structure on the left-hand side, is a component of ribosomes in bacteria, and there's an equivalent in eukaryotic ribosomes as well. The gene that codes for this RNA is about 1,500 nucleotides long, shown in a bar diagram at the top. A portion of the sequence is shown as a DNA sequence at the bottom. Okay? If you look at the bar diagram at the top, you'll see that there are some stretches that are green. The green stretches are variable segments, and there are some stretches that are pink. These are highly conserved segments of the gene, and there is one segment that's black that's invariant in all bacteria. All bacteria have that. Okay? So the basic principle used for using 16S ribosomal RNA to characterize the composition of a genome, of a microbiome, is to PCR amplify 16S ribosomal RNA gene using highly conserved or invariant sequences for the primer design, followed by DNA sequence analysis of various variable segments and comparison to reference databases. Important to realize that what's happening here, you are not isolating individual organisms. You're taking a mass of microbes present in a bacterium, extracting the total DNA. You're throwing in some primers, knowing that these primers will stick to the invariant as well as highly conserved region. So you can now amplify all of those sequences present in the collection. And then you determine the sequences, and then you use computers to align them. We'll show some examples of how it is done in subsequent slides, and to determine the composition of the microbiome in that sample. Okay, so here is here are some details of what actually happens when you try to determine composition using 16S ribosomal RNA sequencing. So from a biological sample, using primers that are targeting conserved regions of 16S ribosomal RNA gene, you determine all of the DNA sequences that come from that PCR step. And then you take that collection of data and you organize them into by sequence clustering programs. In this particular example we have shown a sequence about 50 nucleotides long, and there are 10 sequences here going from 0 to 9. Now, computer programs are very efficient in clustering them, that is, grouping them in their with respect to their relationship with each other. In this particular example, we have used 96% similarity as the cutoff point to define our operational taxonomic unit. What this means is, for a 50 nucleotide sequence, the difference between 1 and 2 is that these two sequences are different from each other. These two sequences are different from each other in two nucleotides because this sequence is 50, right? Okay. So this is sequence clustering, and this is a 96% is being used here as an OTU. Remember that an OTU is roughly the same as species. In real life, the species definition is something like, I think, 98% similarity. I don't remember the exact figure. There's a fractional number. It doesn't matter for our purposes. So after you cluster the sequences, then at 96% similarity, these two sequences, 1 and 2, fall into one OTU, OTU2, whereas this sequence is its own OTU, and so on and so forth. Now, you can graph this to determine relative proportion. In the simple graph, the OTU ID shown here, there's 1, 2, 3, 4, 5, 6, 7. And the relative proportion is shown here. One is present only one copy, so you get fraction 0.1. OTU, there are two members, so it is double the proportion and so on. It is as simple as that. Okay, you can use this information then to place these organisms in the context of the biological world, which is shown on the right-hand side of the diagram, and placing these on the appropriate branch of the evolutionary tree. Okay. In this slide, we show a little bit about the workflow basics of what you'll be actually doing when determining both the composition and the functional capability of a particular microbiome. What is shown in the upper hand part of the diagram is identification of organisms, that is, the composition of a particular microbiome. This is done by 16S ribosomal RNA sequencing. You begin with the sample, for example, a fecal sample, and then you extract the total DNA, and then you subject the total DNA to PCR amplification using as primers oligonucleotides that are complementary to highly conserved portions of the 16S ribosomal RNA gene, and then you cluster the sequences, and then you determine which bacteria are represented in that collection of bacteria within the microbiome. To determine functional potential, you take the same extracted DNA, but this time you subject to shotgun sequencing. Shotgun sequencing means you basically sequence everything that is there. It's a huge mix of things that contains, for example, DNA from bacteria, but also DNA from the host, also DNA from any food sources, everything. A big, huge gamish. But our computer technology and availability of databases very effectively can weed out all DNA other than the bacterial DNA, which is of interest, and then the sequence snippet that are obtained are used to compare with databases to identify what genes are represented in the whole pool. In other words, you try to determine the genetic potential of the microbiome, not specifically necessarily any individual organism and its gene, but the total genetic potential, therefore metabolic potential of that particular microbiome. Okay, all right. Now we come to the topic of how do you express, what are the measures of microbiome diversity? We use two major kinds of measurements. One is called alpha diversity, and the other is called beta diversity. Both are simple concepts. They're illustrated quite well with a simple diagram on the right-hand side that I'm going to walk you through those. There are some other terms here, such as gamma diversity. I'm not going to talk about that. There are other things here, such as Shannon index. I'm also not going to talk about it. So we're going to focus on alpha diversity and beta diversity. So what is alpha diversity? Alpha diversity is simply an OTU count. How many different species of organism are there? It doesn't say anything about how many organisms of the same species are there, simply how many different organisms are there. That is, species richness is alpha diversity. In the diagram, in this particular microbiome at the top, alpha is 4 because there are 4 different species: triangle, star, circle, and orange square. So alpha diversity here is 4. As compared to that, for this microbiome, the alpha diversity is, oops, the alpha diversity is 2. Excuse me while I play around with the placement of, all right, okay. On this side, the alpha diversity is only 2, even though the total number of organisms may be 4, they're only basically 2 species. In this case, the alpha diversity is 3. So this illustration should make it perfectly clear to you. Alpha diversity is simply a measure of species richness or OTU count. So what is beta diversity then? Beta diversity is simply a measure of how different one microbiome is with relation to another microbiome. See, what is the difference between these two microbiomes? That is the beta diversity. If you look at this pair, there are 4 species here. There are 2 species here. However, one of them is the same, right? So you eliminate that. So you between these two, you have 1, 2, 3, 4 species. So that beta diversity is 4. Between these two, beta diversity by the same kind of analysis is 5. Between these two, the beta diversity by the same kind of analysis is 3. Okay. So this is the concept of alpha diversity and beta diversity. Okay. Next, we consider the factors affecting the microbiome. Whenever you see a slide with the stamp illustrative only, that means you don't have to memorize anything on that part of the diagram. It is being presented to you as a resource to look up to satisfy your curiosity. So, and the take-home part I'll summarize in such a slide. Okay. The idea of this slide is that it turns out that the human microbiomes consist of hundreds of species, focusing on the major microbiome, which is the gut microbiome, hundreds of species, about 150 to 300 different types of bacteria in any one individual, but about 1,000 or a few thousand species across all individuals. Okay? And most of this bacteria, at least half, have not been cultured, so we don't know what the species are. We sort of know what big uh, uh, taxonomic phyla they fall into. Okay? So the microbiome varies obviously from individual to individual, but also during different life stages, as well as in response to a large number of host environmental factors, such as diet and disease, underlying disease, and infectious disease, a lot of antibiotic exposure and systemic disease, and a whole lot of other such factors. So this is as you expect it would be. Okay? So in this and a couple of other slides, we deal with the relative bacterial numbers and example taxa and human microbiomes. Again, this side of this slide is stamped illustrative only. That means it's no need for you to memorize all of the different taxa of microbe, just the take-home part. So some of the take-home part is on the left-hand side in the text form. The human organism consists of about 10 to 30 trillion human cells, but the holobiont, though, that is including the microbiome, has at least the same number or slightly larger number of microbial cells, bacterial cells mostly. Okay, and another way of looking at it is that 3% of the human body mass is bacteria. Okay, and as we previously mentioned, microbes or bacteria contribute in something like 9 to 10 million genes compared to human genome, which contribute 20 to 25,000 genes. So just enormous biochemical diversity and capability. Looking at this part of the diagram, the human gut microbiota are dominated by two taxa called firmicutes and Bacteroidetes, but there are many others as well. These are the top six. You don't have to memorize them here, but just be aware that there are some dominant taxa and some taxa are not as dominant. Okay. Now we ask a different question, and that is, where does the microbiome come from? This question can be asked from two different perspectives: evolutionary aspects and developmental aspects. Evolutionary aspects are shown on the left-hand side. And we already know that bacteria were in existence in Earth for more than 3 billion years ago and have always been there, and they underpin the entire biosphere. Even though we evolved late in the history of the planet, we evolved in the midst of microbes. We actually evolved organs to house our microbes. And each body site has its own microbial community, and the evolution of the adaptive immune system allowed for a much richer microbiome than would have been possible. What is meant by this is that adaptive immunity allowed us to coexist with many different kinds of bacterial species without provoking disastrous immune responses towards them. Looking at the developmental aspect, when does the person's microbiome form? From what we know is that at birth, a baby's gut microbiome, which is still not quite formed, is similar to the mother's vaginal microbiome, assuming that the birth has been vaginal. Okay? As you would imagine, a baby born from a cesarean section has a different microbiome that resembles the mother's skin, not the mother's vagina. After birth, the microbiome forms rapidly within the first few years as the baby ingests food. Microbiome in breastfed babies has similarities naturally to its mother's breast and mother's milk. Non-breastfed babies have a different microbiome. In general, microbiome patterns are established in early childhood, and the broad patterns persist into adulthood. Generally, adult individual microbiomes tend to be stable, except for transient changes in response to host and environmental variables. At the end of life, upon death, a completely different microbiome called the necrobiome takes over, and that mediates decomposition, but we are not going to be talking about necrobiome in this series of lecture. On the right-hand side is the diagram that shows an intervention that has been proposed to make a newborn's, a C-section baby's microbiome healthier than it is when you just leave it alone. As we just learned, the microbiome of a newborn born by vaginal birth resembles that of the vagina, but that is considered to be a healthy seeding of the infant's microbiome. But a cesarean baby has a gut microbiome that is derived from the skin, which is considered to be a less healthy version of the microbiome. As a measure to confer the benefit of a vaginal microbiome, something called a vaginal wipe has been proposed, is being practiced in certain birthing centers where the mother's vaginal microbiome is transferred to the baby through a vaginal wipe that is illustrated in the bottom of this diagram. Okay. Let us take a somewhat closer look at the composition and the dominant taxa location and predominant species and taxa of the intestinal microbes. Again, the details are not important. The take-home parts are what I'm going to walk you through. As we mentioned in an earlier slide, the biggest microbiome, the most important microbiome, is the one in the gastrointestinal tract, specifically in the large colon. If you look at the bacterial numbers per gram of tissue or per ml, there is an increase in numbers starting from the stomach going all the way to the large intestine. Okay? The numbers are in the hundreds in the stomach, but they reach billions and trillions by the time you reach colon. The composition also differs, and the composition in normal versus diseased individuals can also differ. It is shown in a overview diagrammatic fashion here. In this part of the diagram on the right hand side, the left side of the diagram is a normal human large colon. Yes, a normal human colon, and this is the colon on the right hand side of a patient suffering from irritable bowel disease, IBD, which is a pretty serious condition. The color scheme here is basically the major taxa. Here, these are the purple is Bacteroidetes, and the green is Firmicutes. And it doesn't matter what all the colors are, but you can see immediately that the color scheme is different between normal gut and IBD gut. And so these kind of differences from the so-called normal is called dysbiosis. That is, the microbiome is different from what is expected in the normal condition. This condition is called normobiosis, and this condition is called dysbiosis. You're going to encounter these terms in subsequent slides. Okay? All right. So the take-home part here is that the major microbiome on the human organism is the one in the GI tract, and the number of bacteria increases progressively from the stomach to the colon, reaching a high of billions to trillions per gram of tissue by the time you reach the large colon. And in a normal individual, there is a particular composition that dominated by particular taxa, and this normal situation is called normobiosis. In certain disease conditions, those the bacterial composition changes, sometimes dramatically, and this condition is given the term dysbiosis. All right, more information on the gut microbiome. On the right-hand side, again, there are lots of details you can safely ignore. It is just for illustrative purposes or to satisfy your curiosity if you want to know more. The information we are interested in is in the text form on the left side. Okay? Some of it is a repetition of stuff I told you already, such as that the bacterial species in a healthy adult number in the hundreds, but not in the thousands, although across all humans, they number in the thousands. Bacteria, DTs, and firmicutes are the major phyla, accounting for more than 90% of bacteria in the GI tract. Bacterial density progressively increases from stomach to rectum, and this roughly parallels food transit times. For example, in the stomach, food stays only 1 hour. In the large intestine, food stays like 35 hours. In the stomach, the bacterial numbers are in the hundreds. In the large intestine, the bacterial numbers are range from hundreds of millions to billions. Right? The abundance of bacterial phyla is determined by 16 as ribosomal RNA species. And one important point I want to drive home here is that colon has very low oxygen tension. So the environment is anaerobic, and therefore the colon is dominated by anaerobic bacteria. In fact, 99% of fecal bacteria are strict anaerobes, a point that you need to remember. Okay, here we come to an overview of microbiome function. There are dozens of important functions, perhaps hundreds of important functions. We are just beginning to mine the information on all the roles played by microbiome. Okay? Some of them are shown here, some important ones are shown here, but this is by no means an exhaustive counting of all the microbiome functions. There are some general functions of microbiome that are given at the top. They serve the function of sealing body spaces. Now, this may sound sort of counterintuitive to you. If microbiome occupies a space, how is it sealing the body space? Well, the idea here is that when there are already occupants in a given body space, the intruders don't find it so easy to gain a foothold. That is what is meant here. They seal body spaces in that sense, and therefore they protect against invasive species from taking root in those spaces. But of course, microbiome has functions way beyond just that. And one of the major functions is immune modulation and maintaining something called gut barrier integrity. What exactly that means will be made clear in a few subsequent slides that are coming down the pipe. Also important is their ability to synthesize de novo, that is from scratch, molecules such as polysaccharide A, which is an important immune regulator. We're going to also come to that in a subsequent slide. Another important role is their ability to digest dietary fibers by fermentation to short chain fatty acids, SCFA. There are three. Those are acetate, propionate, and butyrate. Acetic acid, propionic acid, and butyric acid. Acetate, propionate, butyrate. These are collectively called SCFA. They also have a role in synthesizing water-soluble B vitamins as well as vitamin K. They play a role in conversion of host metabolites, such as primary bile salts into secondary bile salts. They have a role that is presently poorly understood but recognized as potentially important in regulation of fat storage and energy homeostasis. Microbiome is now recognized as being essential, actually, for brain development and behavior. And microbiome is believed to contribute to body heat. Up to 70% of the body heat may be a contribution from microbiome. We'll visit the most important of these functions in subsequent slides. In order to understand the role of the microbiome on gut barrier function, we need first an introduction to the gut epithelium. Okay? The gut epithelium consists of a single layer of cells that line the entire gastrointestinal tract. This, this epithelial layer is crucially important in the interaction of the microbiome with the host, as well as with other functions of the gastrointestinal tract, which obviously includes absorption of food as well as the excretion of several host-derived metabolites, right? Okay, so the gut epithelium is folded into a series of finger-like projections which have the collective function of increasing the surface area of the epithelium. The bottom of these projections, called the crypt, is where stem cells reside. That is where the epithelial cells are continuously produced and exported into the finger-like projection that are the villi. The villi. Okay. The villi contain differentiated cells, but the stem cells which continuously produce new epithelial cells are in the crypt. So cells are born at the bottom of the crypt and they gradually differentiate into various kind of gut epithelial cells, which then they migrate toward the top of the villi. Eventually, some cells die and they are slid, uh, they are uh, they are shed into the lumen and they are replaced by a fresh cell. The entire cycle from birth to death takes about 4 to 5 days. So the gut epithelial barrier is being continuously replenished by fresh cells. Right? All right. So let's look at a few types of epithelial cell. The majority of cells are called enterocytes. They're 80% of the entire epithelial layer. They have a barrier function. They are required for nutrient absorption, but they also act as antigen presenting cells for luminal antigens. For those of you who have not taken immunology before, antigen presenting cells are simply cells that take up pieces of bacteria and then present them in particular forms to other cells that recognize those antigens as foreign bodies and mount a particular kind of immune response. Okay. So they also act as APCs, that is antigen presenting cells. A minority of cells, about 1% of the cells, are endocrine cells, shown diagrammatically um, here as cells lacking the projection fimbriae, right? Endocrine cells, and they produce more than 30 different gut hormones regulating various aspects of gut function, including peristalsis and enterocyte proliferation. Right? About 4 to 16% are goblet cells, shown here. They produce mucus that coats the entire surface of the epithelium. Some cells, a minority again, 0.4 to 1%, are tuft cells that are shown here. They are specialized cells which act as sensors for protozoa and helminths that are present in the GI tract. In addition to that, there are Paneth cells that reside in the crypt and that produce, they're also a minority, they always stay in the crypt, they produce antimicrobial peptides called AMP, such as alpha defensin lysozyme and phospholipase. The important point here is antimicrobial peptides, not necessarily the names of individual enzymes. Okay? What is not shown here is another kind of minor population called microfold cells or M cells. These have the ability to endocytose luminal bacteria and luminal antigen and deliver them to immune cells underneath epithelium. In other words, they are cells dedicated to sampling antigens and bacteria present in the lumen and delivering them to immune cells underneath the below the epithelial layer. Okay, with that primer in epithelial microanatomy, let's go on to our consideration of the relationship between the microbiome and gut epithelial barrier integrity. It turns out that a healthy microbiome is required for maintaining gut barrier, gut epithelial barrier, and in turn, integrity of the epithelial barriers is essential for maintaining a healthy microbiome. This interdependent relationship is shown in a cartoon form here, and some functions that are necessary to maintain barrier integrity and some function necessary for maintaining healthy microbiome are shown in text form here. Examples of bacterial actions that are necessary for maintaining barrier integrity is synthesis of short chain fatty acid. It turns out that short chain fatty acids are the principal source of energy, not glucose, but HCFA, a principal source of energy for epithelial cells. And bacterial cell surface macromolecules such as PSA are required for maintaining gut barrier integrity. Synthesis of bacterial hormones and neurochemical. Bacteria actually synthesize hormones that act on the host. They actually synthesize neurochemicals that act on the host. All of them are necessary for maintaining barrier integrity. Similarly, the epithelial layer affects the microbiome through multiple actions. Some important ones are shown here. One is mucin production that limits access or how close a bacterium can get to the epithelial layer simply by coating the epithelium with mucin. And by producing antimicrobial peptide, this is the function of Paneth cells in the crypt. And by producing secretory IgA antibodies that control the proportion of different bacteria by targeting specific types of bacteria. Okay? So these are all examples of how this interdependent process works. Microbes produce chemicals and produce food for the epithelial cell, and the epithelium in turn produces mucin, antimicrobial peptides, and antibodies that control the population composition of the microbiome.
Dr. Humayun,
Yes,
there's a question in the chat from a student.
yes.
How does the immune system know not to mount an immune response to bacteria in the intestine versus pathogenic bacteria?
Well, we're going to come to that. Excellent question. That's the heart of the matter. And we're going to come to that in subsequent slides. Okay? All right. All right. Now let's ask a different question. Undoubtedly, some of you have thought about this question, and that is, what is a healthy microbiome? Because we already know that there are individual to individual variations, and in the same individuals, there are variations that are transiently occurring in response to diet, in response to antibiotics, in response to underlying disease condition, so on and so forth. And also, in a population, we said there are thousands of different bacteria. In each individual, there are 100 to a few hundred different bacterial species. Under these conditions, how do we identify what's a normal or a healthy microbiome? And how do we reconcile the fact that each individual has a different microbiome and they're all considered to be healthy, and yet when there is a disease, the microbiome, when it changes, that's considered to be abnormal. How do we sort this out? The concept that has emerged is that what is important is the song and not the singer. The idea behind this is as follows. It turns out that even though bacterial composition might change from one individual to individual, their overall genetic potential may be similar because many bacterial species share genes and pathways, metabolic pathways, with similar function. Therefore, you don't need the same exact species to carry out a given function. There are many other alternative bacteria that can also serve the same function. So a healthy microbiome can be constituted in many different combinations, okay? As long as they're singing the same song, that is, they're making the same contributions to the host organism. In dysbiosis, specific beneficial functions may be missing because they're replaced by other bacteria that don't have those functions, or detrimental new functions may be added. In this case, we imagine that the song is off key. Okay? So, so this is the idea. This is how we reconcile the fact that microbiome can be variable from individual to individual, and then variable also within the same individual depending on life stage and life circumstance. We imagine that in a normal microbiome, you have a collection of microbes that together maintain a normal health status by having different combinations of similar functions together. Okay, so in general, a healthy microbiome is characterized by richness, the diversity. In general, a dysbiotic microbiome has decreased richness, that the diversity is lower. Okay, so this is the hallmark of dysbiosis, that is, there's decreased richness. Okay, this and subsequent slides would deal with the question, including the question raised by one of the students, okay, that the role of the microbiome in maintaining immunological equilibrium. What is maintained by what is made meant by maintaining immunological equilibrium is illustrated on the diagram on the right side and in a few words on the left side. In equilibrium, the idea is that the immune system is so finely tuned that it normal symbiont, that is bacteria necessary for the existence of the host organism, and commensals, bacteria that are getting a free ride, but they're not harming the host. But neither are they required for the host to survive. Commensals, symbionts and commensal. Okay, an immune system that can tolerate symbionts and commensals and yet retain the ability to respond to pathobionts if they get a foothold in the system, thereby in inflammatory response. In immune equilibrium, it's possible for the host organism to tolerate symbionts and commensals while retaining the ability to respond to pathobionts if it encounters pathobionts. When there is disequilibrium in dysbiosis, then what happens is that there is a disproportionate, strong inflammatory response to pathogens when they are encountered. Okay? This is the idea of disequilibrium. How this is achieved is a very complex matter, but we'll go through some simple slides to give you an idea of how this equilibrium is maintained in normal, by normal microbiome, how this is affected in the case of dysbiosis. Okay. In this slide, we deal with the question of two different types of responses to two different kinds of ontogenic stimuli in the gut. Okay? Dendritic cells, or DCs, are a class of immune cells which are shown either in this blue form or in red form with a lot of cell projections, like starfish shaped cartoons here represent dendritic cells of two different types. And in the diagram, here is the gut epithelial barrier. This is the lumen. The lumen has the gut bacteria as well as other organisms. And under the epithelial cell is the various components of the immune system. Okay. In normal, in normal, when there is a normal microbiome present, which is dominated by commensals and symbionts, the antigens present in the lumen are sensed by a class of DCs called tolerogenic DCs, the blue DC shown here, and these antigens are presented to the immune system underlying the epithelial barrier to activate a tolerogenic T cells called Treg cells, which ultimately produce a, a tolerogenic cytokine. A cytokine is a small molecule. A cytokine is a signal to other cells to carry out particular function, sort of like a cell-to-cell hormone, right? So Treg cells will produce a signaling molecule called interleukin 10, IL-10, that basically prevents an inflammatory response from taking hold in response to these antigens. So, but certain other DCs that are present also under the epithelial barrier are designed to sense pathogenic antigens, such as those produced by harmful bacteria. When those antigens are detected, these DCs produce a different interleukin called IL-23 that triggers a strong immune response sufficient to clear those antigens. This is the normal situation. In normal cells, inflammation is kept in check because most bacteria in normobiosis, in a normal microbiome, activate mostly this pathway that leads to production of IL-10 that keeps inflammation at a low level. Okay? It is only when this is disturbed so that there is more of a pathogen that is present that a strong immune response will be, a strong inflammatory response will ensue. Otherwise, the inflammatory response remains low. This is the setup. In the next slide, there are some greater details using one specific bacterial antigen as an example. As an example, this is an important antigen. Okay, that particular antigen is called PSA or polysaccharide A. Is also called exopolysaccharide A. PSA is shown here in these wiggly diagrams. This is basically a surface molecule, a piece of a polysaccharide with some other components in it. Okay? This is a very important molecule produced by an organism called Bacteroides fragilis. It's very important in training bacteria into a tolerogenic response. That part, what happens in normal cells, is shown in the middle part of the diagram. Here, a dendritic cell that sends PSA will present PSA to CD4 T cells, which ultimately result in activation of Treg cell and production of IL-10. IL-10 is the signaling molecule that keeps inflammation low, blocking inflammation. Okay, all right. It turns out that PSA is very important to the host organism, not only because it blocks inappropriate inflammation, but IL-10 can also travel to other parts of the body and keep systemic inflammation low as well. That is shown here. It can travel through the bloodstream and the lymphatic stream and reach distant organs, for example, CNS, and keep inflammation low. It turns out that inflammation underlies a whole lot of human disease. Cardiovascular disease, CNS diseases, all sorts of diseases are triggered by system-wide inflammatory conditions. So PSA plays a role not only in the gut itself, but by producing IL-10, which reaches distal parts of the host, also controls inflammation in other parts as well. Please excuse me for one moment. What is also shown in the diagram on the left-hand side is another way of showing the importance of PSA and PSA-like molecules in training the immune system. What is shown on the left-hand side is the situation in germ-free animals. In germ-free animals, there are no gut microbes. In this condition, the immune system is not well trained, is not in equilibrium. Okay, it's only when bacteria are present in the gut that the immune system reaches equilibrium, and the equilibrium is reached through the mechanism that we discussed in this slide and the previous slide. Okay, now we move on to a second important role of the gut microbiome, which is the ability to digest food fiber and the ability to regulate energy homeostasis. Okay, you already know that humans lack enzymes to efficiently digest many plant-derived polysaccharides. For example, a component of starch, so-called resistant starch, is not digestible by human enzyme. Also, the so-called dietary fiber. Dietary fiber basically is plant lignin plus plant cell wall component such as cellulose, pectins, inulin, etc., etc. These are not digestible by human enzyme. However, these are the principal substrates on which intestinal bacteria, specifically large colon bacteria, exist. They basically eat the dietary fibers. Okay, the product of utilization of these fibers, they are utilized by fermentation, because remember that there's no oxygen here, these bacteria are anaerobic, right? So they can only ferment carbohydrate. So the product of fermentation are the SCFAs, acetate, propionate, and butyrate, shown here. Acetate, propionate, and butyrate, right? And they're made in a particular proportion. Acetate is dominant, and the others are in equal proportion, propionate and butyric acid. Right? Now, a degradation of the fibers is mostly carried out by members of Bacteroidetes, which is a very large portion of the gut bacteria. Some particular bacteria have been studied well, such as Bacteroides theta iota micron. Excuse me. All right. A few words about the significance of SCFAs. We know that SCFAs are significant because of a number of clues. They play an important role, we already know, in the gut, because they are the energy source for gut epithelial cells. But they also have other functions in the gut. But most importantly, SCFA receptors are found throughout the host tissues, implying that SCFAs must act on distal organs and distal tissues away from the gut itself. Some of those sites are shown here in diagrammatic form. You don't have to remember all of those, and particularly the pathways you don't have to remember, but remember that there are tissues such as organs such as brain, peripheral nerves, white adipose tissue, WAT is white adipose tissue, white fat, brown adipose tissue or brown fat, gut, blood vessel, liver, muscle, all of these organs have receptors for SCFA, meaning that SCFA produced in the gut by the microbiome can reach and affect and have an influence on how all of these organs or tissues operate. Right? SCFAs are recognized to have broad anti-inflammatory effects, but through what pathways is not yet well understood. Okay? There are certain hypothesized roles for SCFAs in energy homeostasis, And those roles include a role in promoting as well as controlling obesity. Okay, so they can promote both obesity as well as anorexia. Obviously, these are opposing traits, but it seems like SCFA are involved in this balance. The exact mechanisms are not known, and SCFA might also increase insulin sensitivity. Obviously, this has an effect on chronic conditions such as diabetes and metabolic syndrome. Now, we already know that gut microbiota produce SCFAs, but what we have not seen until now is that gut microbiota also produce a whole lot of other bioactive small molecules. Some examples are shown here. They produce antimicrobials, that is, warfare against other members of the microbiome. Antimicrobials such as RIPS, antibiotics, bacteriocins, heat stable enterotoxin, many antimicrobials. They also produce hormones and neuroactive chemicals. These act on the host. They include indole, imidazole propionate, gamma aminobutyric acid, for example. They also play a role in immune priming and neuronal signaling. For example, we already saw the role of polysaccharide A on immune priming. Right? All of these are produced by gut microbiota. There are additional roles of the microbiome. Two of them are illustrated here. Again, the details are not important. The take-home parts are important. On the left-hand side, the take-home part is the following. Gut microbiome produces significant portions of B vitamins. The B vitamins are listed here by name, and the daily recommended uh, um, intake values are shown here. You can see that many B vitamins, such as cobalamin, folate, niacin, are a significant contribution comes from microbiome. The remainder comes from food. For example, pyridoxine B7, almost 90% is produced by microbiome. The remainder comes from food intake. So the take-home part, this part of the slide, is that gut microbiome produces a lot of vitamins, especially B vitamins, but also vitamin K that's not shown here. On the right-hand side is the role of microbiome in modifying host metabolites with specific focus on bile salts. What is shown here in diagrammatic fashion is the bile salt circulation system, sometimes called hepatobiliary circulation. In very simplified terms, what the system is the following. Liver makes compounds that are derived ultimately from cholesterol backbone called bile salts and secretes them into the gut. And in the gut, they are reabsorbed from the gut and delivered back into the liver. This is the so-called bile salt or hepatobiliary circulation system. Okay? While the bile salts are in transit through the intestine, some of them are acted upon by microbiome present in the gut, and their chemical nature is modified. Some of those chemical changes are shown here. You don't have to memorize them. Just know that some of the chemical structures are modified to create new kinds of bile salts, which are then reabsorbed and delivered to the liver, where they exert specific metabolic consequences. Now, it is suspected that this bile salt modification plays important functional roles, but what those are is not yet determined, but it's suspected that they have important functional roles. So, all right. In this slide, we'll look at another aspect of the microbiome, and which is that, as you would suspect, oral medicine that are must be affected in some fashion by the microbiome. of course, it is affected. Oral medicine, as well as a group of chemicals that we call xenobiotics, okay, are affected by the microbiome. They're affected both in obvious ways, what you intuitively would guess is possible, but also by a pathway that you may not intuitively get. And some of those concepts are shown in this in a diagrammatic fashion here. So, oral medicines or xenobiotics in general, you will expect may alter gut microbiome composition. Obviously, this will be the case for antibiotics, but it may also be the case for many other kind of chemicals. They may alter microbiome composition. In turn, those xenobiotics, whether they're medications or others, will likely be affected by the microbial enzyme. For example, an active chemical, an active medication may be converted to in an inactive form by the action of the microbes. Right? Similarly, an inactive xenobiotic, inactive chemical might be activated by the action of the microbial enzyme into something that exerts an influence on the host organism. Both of these are intuitively obvious. What may be less obvious to you is the possibility that by changing the gut microbiome may affect host metabolism, the host metabolism of the xenobiotic itself. That is, after it is absorbed, what happens to that xenobiotic in the bloodstream may be affected by the gut microbiome acting on host metabolism. And of course, the presence of xenobiotic might also change, the presence of microbiome might also change the absorption of the xenobiotic into the gut. So, what is clear, the bottom line of this slide is, of course, the microbiome has a strong influence on ingested pharmaceuticals and other xenobiotics. Okay. Okay, so we're going to move on into the next part of this lecture, which is the role of microbiome in human disease. We are going to take up this matter in the second half of the lecture coming up next week. So we are concluding our lecture for now, and I'm going to close my slides, and I'm happy to answer one or two questions if you have them. I'm going to scan the chat now to see if there is any new questions. I don't see any new questions. So we are done for the day. We'll catch up with the remainder next week.
Zachary, don't forget to stop the recording.
Oh, yes, important. Yes. And what do I do next after that?
Don't do anything. It'll just process. You'll get an email once it's been processed. It'll just sit in your Zoom account, and I probably won't need it. But if I do need it, it'll be sitting there and we can extract it if we need it. Okay.
Okay, everyone. Good night. Bye, and see you next week.
Thanks, Zachary.